Hi friends, I hope you all are preparing well, and today I have come with another topic which is seed and bud dormancy. So this is another concept building video where I will discuss some questions based on seed and bud dormancy. So the first question is, what is seed dormancy? So we all know what is a dormant stage, right? What is a dormant stage? It is a resting stage where no growth is seen. Okay, the seed is still alive, but no growth is there. It is in the resting condition, but seeds are still alive. So this seed dormancy, it is a living condition or state in which there is no growth. Okay, in which growth does not occur. Until what period will the growth be stopped? So the growth will be stopped until the favorable temperature returns. Okay, so growth will not occur under conditions that are unfavorable. So under the favorable condition only the seeds will grow, and when the environment is not favorable, the seed becomes dormant. That means it is still alive but in the resting stage. Okay, next is, what is scarification? What is scarification? That means the best example you can take about gram seeds. Okay, they will not germinate but they are still alive. When they get the water, when you imbibe the gram seeds, they show germination. But when you buy it from the market, they are very dry. That means, are they dead? No, they are still alive, but they are in the dormant stage because the environment is not favorable for their growth. So under what condition will the environment become favorable? When they get lots of water. When the seed soaks water, the seeds starts germinating. That means they start showing growth. Next is, what is scarification? So there are several processes by which we can promote the process of seed germination. So scarification is among one of them. So this scarification, it is a process of seed germination. It is a process of seed germination. What happens in this process? In this process, the seeds are mechanically disrupted so that seed coat can be removed. So seed coat is a hindrance for the seed to germinate. They are very hard. Okay, the plumule cannot come out from there. Okay, so see here. So in this process, the seeds are mechanically disrupted. Mechanically disrupted. Why? What is the motive of disrupting? To remove the seed coat. Seeds are mechanically disrupted to remove the seed coat. Clear? So main keyword here in this question is, in scarification, the seeds are mechanically disrupted to remove the seed coat. Next is, what is hilum? So if you have seen any seed, say for example I take the gram seeds only. Gram seed, you must have seen that there is a small pore. There is a small pore like structure. Okay, small pore like structure which is the route of entry of the water. If you will put this gram seed into water, through this pore only the water will enter in. Okay, so this is a pore. This is a small, this hilum is a small pore, and this is the only possible route of entry. or exit of water. The seed will not absorb the water from throughout its whole surface, but through the hilum, the water will take entry into the seed. Okay, so what it is doing actually, it is controlling the seed moisture content. Okay, and they are found in many seeds, including example, they are found in seeds of leguminous plants. Like for example, pulses family of plants. Next is, what is strophular plug? So remember, there are many seeds which have pores, and these pores are blocked with a plug called strophular plug. So I said that many seeds they have pores, and these pores are blocked with a plug known as strophular plug, and this plug has to be mechanically removed or dislodged before the water and oxygen can enter. So what I said, many seeds have pores that are blocked with a plug, and this plug is known as strophular plug, and which has to be removed mechanically. What for they need to be removed? Mechanically dislodged. The appropriate term is dislodged before water and oxygen can enter. Water and oxygen can enter. So this plug is basically, it is blocking the entry of water and oxygen into the seed. So we have to dislodge this plug to allow the entry of water and oxygen. Next question is, name few inhibitors that cause the seed dormancy. So first is a plant hormone which causes seed dormancy is abscisic acid. Next is, there are few unsaturated lactones are also there. Unsaturated lactones, which is coumarin. Next is, there are some phenolic compounds which also cause seed dormancy. Example is ferulic acid. Okay. Next is, what is thermoperiodism? So this thermoperiodism, it has been best explained by taking the example of tomatoes. So tomatoes, they grow very poorly when they are kept at either 26 degrees centigrade or 18 degrees centigrade. Either 26 degrees centigrade or 18 degrees centigrade. But when the condition is changed, say for example, under alternation, temperature such as 26 degrees centigrade at day, but when, just a moment, but when they are provided with, they are given 26 degrees centigrade at day and 18 degrees centigrade at night, then this condition causes vigorous growth and maximum production. So this is the temperature effect. Okay, so this temperature, if this temperature cycle alternates the growth is even poorer at 26 degrees centigrade. So if this cycle, that means if they are kept at 26 degrees centigrade alone, or if they are kept at 18 degrees centigrade alone, then the result will be even poorer. Okay, so thermoperiodism is, by taking the example of tomatoes, it has been explained that they grow at constant temperature either at 26 degrees centigrade or 18 degrees centigrade, but they grow poorly at this temperature. When they are provided with 26 degrees centigrade at day and 18 degrees centigrade at night, then they causes vigorous growth and maximum production. If this cycle alternates or changes, then the growth is very poor. Okay. Next is, how does dormancy differ from quiescence? Now quiescence is a very important term in this chapter. What does this mean? It means that, first of all, what is dormancy? Dormancy, I already explained. It is a condition, it is a resting condition of a seed when the seed fails to germinate because of some internal condition, maybe because of some external condition. Okay, so when seeds fail to germinate because of internal or external condition. What are the external conditions that can cause the seed to remain dormant? Say for example temperature, then moisture, etc. Okay, and when the environment is suitable, then only they will germinate. But what is this quiescence? So quiescence is, it is a condition of a seed when the seed is unable to germinate only because of external conditions, only because of suitable external conditions are not available. Okay, so I write down, it is the condition of a seed when the seed is unable to germinate only because of suitable suitable external conditions are not available. Okay, and under what condition? Say for example, if I take, if the seed is very dry, or if it is too cold, in that condition the seed cannot germinate even when the internal conditions are favorable but external conditions are unfavorable. Okay, next is, what do you mean by paradormancy? So this paradormancy, it is another term for dormancy that is controlled by plant parts other than the one being considered. That means if this is a plant, okay, if this is a bud, if this is a bud, okay, and these are the nearby leaves. These are the nearby leaves. So because of the nearby leaves, this bud are not able to germinate. Okay, so this paradormancy is another form of dormancy in which the plant parts other than the one being considered. So here which one we are considering? We are considering the bud. So other than the bud, that is nearby leaves. So nearby leaves, they are not allowing this bud to germinate. Okay, so this dormancy, paradormancy, it is another form of dormancy. What does this mean? Here, this dormancy, it is plant parts, controlled by plant parts other than the one being considered. For example, dormancy of bud controlled by nearby leaves. Next question is, what is impaction? So sometimes when the seeds are vigorously shaked, in that condition the strofular plug is dislodged. This treatment is known as impaction. Okay, now this is also the process of removing the dormancy. So sometimes upon vigorous shaking of seeds, dislodges strofular plug. And this kind of treatment is known as impaction. Okay, in which seeds can this process will be performed can be performed they can be performed in seeds of Melilotus alba, and there are many more. Okay, next is, what is bud? So the bud term you must have heard, but what is the scientific definition of bud? So bud is actually a meristem. It is actually a meristem. So what is how it has been formed? So it is a meristem in which the internodes have failed to elongate. In which the internodes you must remember that these are the elongated regions, and nodes are the swollen region from where the leaves and fruits and flowers emerge. Internodes are the long spaces, okay, in between the nodes. Okay, so in which the internodes have failed to elongate, and the leaves have not yet enlarged. So this structure is enclosed in a set of modified leaves. Okay, so what we see that the buds, they are enclosed by the set of modified leaves, and these leaves are known as what? These leaves are known as bud scales. Okay, so they are enclosed in a set of, just a moment, in a set of modified leaves which are known as bud scale. Okay, so this is for today's class. I hope this has helped you. Thank you so much.